Hello everybody. We finished with what we normally call the metabolic part of 482, and now we will start with the part of Biochem 2 that is focused on molecular biology. Even though the title of chapter 25 is DNA metabolism, indeed that term is technically correct, the topics that we will discuss from now on are more characteristically described as molecular biology rather than metabolism. So in this chapter, we will talk about specifically DNA replication and some of the principles, fundamental principles involved in DNA replication, and then we will also look in a little bit more in details, but let me not get ahead of myself. So I will start from the very beginning. And that is that nucleic acids are carrier of genetic information. More specifically, DNA is the storage of the genetic makeup or information for a given organism. Each one of our nucleated cells contains an exact and the same copy of DNA that we've had when we were first conceived, in that first cell that we began from. How is it possible that genetic information, billions of bases, have been copied thousands if not millions of times without errors? Now, by saying that without errors, the same DNA, that's a relative term. Obviously, there are small mistakes that accumulate, that happen over time, but in general, the percent of these mistakes is so small that it's negligent, unless it hits something that is critical, with critical importance, and then we can obviously observe disease happen. But in general, the principle is that from the moment we were a single cell until now, there have been countless divisions, and the genetic information have been copied fairly consistently and quite securely without significant number of errors, as you will see a little later when I talk to you about the errors in replication. Well, in principle, that is possible because replication uses a template. And now we know that the template is the mother or the old strand of double-stranded DNA, and one of the old strands is used as a template to synthesize a new hybrid, meaning made out of an old and a new strand of double-stranded DNA. That method of replication is known as semi-conservative. And again, that is because of the two new strands of the newly synthesized DNA. One of the strands is from the template or old DNA strand, and one of the strands is newly synthesized. So a big reason why we can copy DNA almost perfectly so many times is because we use a template. Well, that is a good reason, but it's not the only reason. Another reason is also because the synthesis of DNA follows this exact prescription that each A in the template will associate with a T, and each T in the template will be associated with an A, and the same goes for G's and C's. Each G with a C and each C with a G. So together, the fact that we use a template and the fact of the well-known base pairing we can multiply DNA countless times with almost no error. Okay, how was that though first discovered? It's actually a quite ingenious and extremely simple process or test, and that is described in this figure. So what did the scientists at the beginning do? Well, first, it's shown here in A, they simply grew bacteria in media and broth that contains heavy nitrogen, nitrogen 15. And they allowed that bacteria to grow there for a long time, several generations, to a point where basically almost all the nitrogen in the bacterial culture was heavy nitrogen, or nitrogen 15. So they harvested the DNA from the bacteria and they put it on the cesium chloride density gradient gel, and they looked where the DNA positioned. Then they took these cells and they placed them in media that contains lighter nitrogen, for example nitrogen 14, and they allowed only one growth cycle, basically the bacteria to double, to divide only once. And then they harvest the bacteria, they lysed them, and they pooled the DNA and run it again on a cesium chloride density gradient gel. And interestingly, they saw a band that was higher than the original with the heavy nitrogen band, suggesting only one product. Now, if replication was conservative, meaning that the old strands were used as templates to generate new strand, and that each new strand is made only by newly synthesized DNA, then this would not have been the case. We would have still seen the original and the new one. But that's not what they saw. And seeing this strongly suggested that replication is a semi-conservative method. In other words, each newly synthesized DNA double strand is made by one old and one newly synthesized DNA strand. Then they allowed the cells to divide one more time. They took the cells, they lysed them, and they looked at their DNA, and now they saw two products, which again supported the first observation. Why is that? For example, if you take this DNA and you use each one of those strands as templates, the heavy strand will produce a hybrid strand again that contains heavy and light nitrogen. But the light nitrogen strand, which was generated in this case, serving as a template, will pair with another light nitrogen strand, giving rise to this other band of DNA on the cesium chloride gel. And so you can see how this extremely simple experiment proved or gave the scientists a great idea how the replication actually happens. Well, just a general idea of how it actually happens. Or the fact that each old strand of DNA, or parent strand of DNA, is separated into two, and each of the two serves as a template on top of which a newly synthesized DNA was produced or was paired with. Alright, so to understand how replication actually works, the scientists needed to answer these questions. So we need to answer these questions. When does it happen? Where does it begin? And how does it happen? Well, obviously, when do we need to double our genetic information? When there is a cell division. So that was kind of a All right, easy question to answer. Now, what drives the cell to divide? That's a very complicated question, but if we need to have two cells, obviously we need to have two pairs of the same genome if they were to function properly. And DNA replication begins at a special sequence known as the origin of replication. And we're going to talk about that a little later, or named ORI. And the replication moves in both directions from the origin. In other words, we always see the two replication forks. Now, the toughest question is how replication actually happens. What drives it? What controls it? What is the mechanism? This is a more complex question. And by now we already know that DNA synthesis always moves in the 5' to 3' direction, and it's semi-discontinuous, speaking about both strands of DNA. Now, if the synthesis is always done in the 3' to 5' direction, which basically means that the new nucleotides are always added to the 3' end of the growing chain, then the template must be read from 3' to 5' direction. In other words, the replication goes in this direction. It reads the template 3' to 5' and synthesizes the new DNA from 5' to 3'. And we'll see why that is a little later, chemically speaking. So can both strands be synthesized simultaneously and continuously? Well, by now you already know that the answer is no. And that is because the two strands are anti-parallel. If the sense strand is 3' to 5', and the reading happens in this direction, and the synthesis of the newly generated DNA strand is continuous, that's not the case with the antisense, also called the lagging strand. So this is the leading, this is the lagging strand. The lagging strand is 5' to 3'. That means that it will start somewhere and it will go in this direction. But you will see how the double strand is split, and while this will continue, this part though, if we don't continuously start new initiations, if I can use that term, of replication, it will remain single-stranded. These fragments that are basically formed as a result of the transcription of the lagging strand are known as the Okazaki fragments. So the lagging strand is replicated in pieces which later on are ligated and combined together into one strand. And these fragments again are known as the Okazaki fragments. The DNA synthesis is done by an enzyme known as DNA polymerase. I'm sure everybody knows this by now. So, an interesting fact in the synthesis of DNA is that the nucleotide polymerization by itself has a very small free energy change. Then what drives the reaction? What makes DNA be replicated? Alright, you can see right here and even right here what is the chemistry or the mechanism of the linkage of the nucleotides with the growing chain. You can see how the oxygen from hydroxyl group on the 3' carbon attacks the alpha phosphate of the new nucleotide, and this reaction is driven by nucleotide stabilization, by base pairing and base stacking. You can see the stacking and the pairing here, but also by something that, if you look carefully, you already would realize where am I going with this. Pyrophosphate is released. It's an excellent leaving group. But what do we know about pyrophosphate when it's released in the environment? It is highly unstable, and it will be degraded almost immediately into inorganic phosphate, which makes this reaction irreversible because one of the products cannot easily be taken back and returned to the monophosphate nucleotide to form triphosphate nucleotide. That fact itself renders this reaction irreversible in following this mechanism. I also want you to notice the magnesiums as part of DNA polymerase. If you remember, if you're taking the DNA lab, or if you have taken the DNA lab, and if you also remember our discussions in 481 when we talked about PCR, magnesium chloride or magnesium sulfate, magnesium ions basically, are of critical importance for the success of a PCR reaction. And that is because they're part of the active site, and they help the positioning of the nucleotide in a specific orientation, interacting with the negative charges of the oxygen on the phosphate tail of the newly coming nucleotide, and also drawing some of that electron density towards themselves to allow this reaction actually to happen. So the magnesium ions are of critical importance for the success of DNA polymerization. All right. Now let's focus a little bit on the DNA polymerase functionally. We're going to talk about it structurally next time, but functionally, what is required for DNA polymerase to work? So in addition, for polymerization to happen, we need nucleotides and the right environment. Okay, everybody will agree with this. But DNA polymerase also requires a template, and it requires a primer with a 3' hydroxyl group. Someone might say, well, do we have primers without a 3' hydroxyl group? Well, if we want to synthesize it without a 3' hydroxyl group, we can. So most obviously in nature, all primers or all 3' ends will have the hydroxyl group, but when we do this in vitro, it doesn't need to have the hydroxyl group. We can synthesize one without, for whatever reason we want to use it for. Anyway, what I'm highlighting here is the hydroxyl group, because if you remember, that is the group that will be doing the attacking, making possible the addition of new nucleotides. Different DNA polymerases have a different rate of additioning nucleotides into the growing chain. I'm asking you here, I don't know whether you remember, but what is the rate of nucleotide addition by DNA polymerases used in the PCR reactions? For those of you that have taken the DNA lab, hopefully you remember that, a safe estimate could be 1,000 bases per minute, but the actual rate is a lot faster. However, in an in vitro environment, usually it goes between a few hundred and 1,000 nucleotides per minute. Another term that describes DNA polymerases is processivity. Processivity, by definition, is described by the amount of nucleotides that a DNA polymerase adds before it detaches or falls off the template. In other words, you can think of it as almost as stamina in a way. How long can you run without taking a break? For a water break, or to catch your breath, or sit down and rest a little bit. That's basically processivity for DNA polymerases. How long can DNA polymerase continuously add nucleotides before it detaches or falls off a template? So DNA synthesis is like I said, a very accurate process, but there's something very interesting about that accuracy, and that is it's been found that wrong nucleotides enter the active site with a frequency of 10 to the 4th, 10 to the 5th. In other words, for every 10,000 correct nucleotides that enter the site, or every 10,000 to 100,000 nucleotides that enter the active site, we get one that enters wrongly. But wrong nucleotide additions in the newly synthesized DNA molecule are observed with a frequency of only 10 to the 9th, 10 to the 9th, 10 to the 10th. That's 1 in a billion, 1 in 10 billions. In other words, if you think about it, for a human genome, you're going to see for one replication possibly one error. I can't remember exactly how many nucleotides were there. For some reason, I think that our genome is about 3 billion bases. So if that's the case, you may not even see a mistake in a single replication of our entire genome. That is how sensitive and accurate this process is. But why the difference? Look at the difference. I mean, if really there's one wrong nucleotide in the active site for every 10 to 100,000 base pairs, this number should be a lot lower. But instead, it is so much higher. I mean, double if you think about compared to this. Instead of 1 in 100,000, now it's 1 in 10 billion errors. So what is the reason for this accuracy? Well, one of the reasons is base pairing, which is a good reason. But I usually would ask you here, for free nucleotides, I'm talking about if we have free nucleotides in solution, can they base pair with any other free nucleotide in the solution? The answer to that is yes, of course. A's can base pair with T's, G's, and C's. And so do they. Each one of them can base pair with all the others. Why? Because they're free, and they can achieve any orientation they want. But that's not what happens in the active site of DNA polymerase. The geometry that the pairs achieve is critical for them to actually work out. If you have a T in the active site from the template, only A will fit in the active site's parameters. If you have a C shown here, only G will fit in the active site's parameters, and vice versa for both cases. But if you have a G and you try to fit an A, that won't happen very well. It will not fit well into the active site. Same thing if you have a C and you try to fit an A, or a T and you try to fit a G. So you can form these, technically speaking, theoretically speaking, but the active site, or more importantly, the geometry of the active site, will not allow for these base pairs to occur. Only T and A and C and G will fit within the geometry or the structure or the 3D orientation of the active site. So for that reason, this process alone is so precise. Now, it's not the only reason though. There's another reason that is also very important, and that is DNA proofreading. So how does DNA proofreading works? Well, strictly speaking, when we talk about DNA proofreading, that means the 3' to 5' proofreading ability of DNA. In other words, suppose, as is shown in this figure, that you have a polymerase that is adding nucleotides. And at some point, the wrong nucleotide, shown here, C, comes in to bind with an A. Well, obviously, that's not going to happen because it will be odd for that to be fit in there, and the A and C will not pair very well. But let's say that the bond, the phosphoester bond, was made already. And when the polymerase wants to move on, the fact that this base pairing doesn't work properly will halt the DNA polymerase, and it will force it to go back. And when it goes back, the active site for the proofreading will be on top of the mispaired nucleotides, and the 3' now to 5' exonuclease activity, exo because it cuts out DNA from the end, it will remove the wrong nucleotide, and then it will add, and then it will continue moving forward, and it will add the right nucleotide in that place. Now I'm asking here, is this just a reverse reaction or not? And the answer is no, it is not the reverse reaction. And if you remember, why is that not the reverse reaction? Because of this pyrophosphate. Once generated, it almost instantly gets degraded, so we no longer have it available for this reaction to go back. So that's why we need a different active site, as shown here, the 3' to 5' proofreading or exonuclease activity site that will excise the wrong nucleotide and then allow the polymerase to move forward and synthesize the rest of the DNA and add the correct nucleotide in its own place. Now there are several different DNA polymerases, 1, 2, 3, 4, 5 shown here. The one DNA polymerase that is primarily involved in the genetic information replication, it's the Pol3 or Pol-C polymerase. The others have other functions which are usually involved in DNA repair. You can also see right here the polymerization rate for Pol3 is 250 to 1,000 nucleotides per second. I'm not talking about per minute now. We're talking about per second. That's how fast things are moving in the cell. And notice the others. 60 to 20, 40. I mean, how long will it take for Pol2 to transcribe or to replicate the entire genome? Well, 200 times less according to this image. So it's going to take an awfully long time to replicate any cell if we use this rate. Also notice that they all have 3' to 5' hexanuclease or proofreading ability. An interesting thing though, there is another exonuclease activity that some polymerases have, and that is the 5' to 3' exonuclease activity, or that's a forward exonuclease activity. Only polymerase 1 or Pol-A has that activity. I wanted to ask you here, why wouldn't you want to have 5' to 3' exonuclease activity on Pol-C? Think about it, especially in the sense of the Okazaki fragments. If you have a polymerase basically going 1,000 nucleotides per second forward, and it reaches the beginning of the previous Okazaki fragment, what's going to happen if that polymerase has 5' to 3' exonuclease activity? It's going to plow through, and by the time it realizes what's doing, you're going to have one or a few thousand nucleotides being removed. In other words, it's not an efficient process. We're going to just waste a lot of energy to simply synthesize and then degrade it because of a 5' to 3' exonuclease activity. For that reason, this is my speculation of course, it makes sense, Pol3 does not have 5' to 3' exonuclease activity. We have a different polymerase, and that is Pol1 or Pol-A, that has much lower rate to allow more finite and slower addition, and also it can remove nucleotides at a slower rate. This is shown on this image. Like I said, Pol-A or DNA polymerase 1 has that ability, and like I said, it's used in DNA repair. Each Okazaki fragment, we're going to talk about this later, is started by an RNA primer. But regardless of whether it's RNA or DNA, the cell usually tries to remove this primer, especially if it recognizes something foreign in a nucleotide. This case mostly plays a role with the RNA primer removal. That's the starting point of Okazaki fragment. DNA polymerase comes in, starts excising the RNA primer or the RNA nucleotides until it reaches the DNA nucleotides. Then it stops and falls off. And now we have a repaired segment free of RNA and a nick. This nick, we'll see later, will be repaired by a DNA ligase, and it will be like it never has been there. All right, well, I think that is pretty much I wanted to talk to you at this point. Next time, I will continue with the structure of DNA polymerase and the stages of DNA replication. All right, thank you for your time. And again, if you have any questions, please let me know. Thank you.